Thanks, Steve. It's really great to see all that come together. Very exciting times. So it's my pleasure now to introduce Mary Dickinson. Not only is she a truly amazing developmental biologist, but she also happens to be the current Associate Dean for Research at Baylor. I'm not exactly sure how she does both of those things at the same time, but today she's not going to talk to us about her dean duties. But her lab uses some really innovative multidisciplinary approaches that combine mouse genetics, developmental biology, imaging, and bioengineering to really precisely model human disease. And she's also, as Steve mentioned earlier, involved in the early developmental assessments in the IMPC. So she's going to talk to us about both of those things today. So thanks so much for coming, Mary.
Thank you so much. Thank you so much. So Steve provided a wonderful overview of the IMPC program, and I'd like to now focus on the analysis of essential genes and looking at developmental phenotypes. So this is the slide to sort of pick up where Steve left off about the overall progress. I won't dwell on it too much because you've just heard this information, but incredible inroads are being made into generating null alleles. And there are a variety of pipelines established to look at adult phenotypes and now late onset phenotyping, and to provide rich and robust information about pleiotropy, as Steve alluded to. One of the things that we developed a couple of years ago is a pipeline dedicated to looking at the 30 to 35% of mutations that are recessive lethals. These yield, in order to characterize these phenotypes, we had to build a pipeline that was really dedicated to understanding embryonic phenotypes. And that's what I'm going to discuss with you today. Part of the analysis initially is to establish primary viability. And through the establishment of primary viability, we then have three outcomes that come from that analysis. Mice are either viable as homozygous nulls, or lethal, meaning that there are no homozygotes found at weaning, or they fall into an in-between category that we term subviable. And these mice, these lines are characterized by a lower percentage of homozygotes found at weaning. In our case, the threshold is less than 12.5% homozygotes found at weaning. And these subviable alleles occur at a frequency of about 10% overall, whereas the viable alleles are at 65% and the lethal alleles occur at about 25%. These percentages, as I'll allude to later, are very highly reproducible. And looking at the null alleles through the IMPC efforts, comparing them to ENU screens and other mouse mutagenesis efforts, these numbers are really highly robust and repeatable over a large number of different groups. So essential genes, I want to talk about the role of essential genes. Essential genes have been described in a variety of different circumstances, and essential is a general term, and it really lends to definition for individual experiments. So some people consider essential genes needed for cells to survive in a dish. In our case, we are characterizing essential genes as those needed to produce a viable animal. So for instance, our lethal and subviable lines that don't survive to birth or weaning we consider those essential genes. But you could also think of essential genes as anything that would reduce the life expectancy of an animal. So there's been a variety of different ways that people have considered this, but we'll focus on the middle term. So to evaluate the phenotypes produced in these animals, we established a pipeline to investigate development at a variety of different stages. So we have the opportunity in the mouse to look, we chose 4 different stages to focus on, and they each have different hallmarks associated with different aspects of development at those stages. So first, if we look at E9.5, which is a relatively early stage in development, homozygous embryos that don't appear at these stages have already perished and are probably due to defects in either pre-implantation development or early post-implantation development. And the figure that I show here shows what mouse embryos look like during those stages. So very early defects, gastrulation or organization of the body, plan defects, or the very beginning of heart formation. Whereas when you look at 12.5, the types of defects that you find in those embryos, oftentimes if lethality is found at that stage, it's generally due to poor heart function or poor circulation, placental formation, and other types of defects related to the very beginning of the heartbeats, because those are the stages between 9.5 and 12.5 when that is all beginning to come online for the embryos. And this just shows a diagram of the extra embryonic tissues that we consider in our analysis, and also the general overview of what an embryo looks like at this stage. Further in development, defects that cause lethality at these stages are often related to defects in more later stages of organogenesis and also heart failure. So not just the development of the heart or the circulatory system, but actually the continued functionality of that system. And many lines are viable at 18.5. We find in our analysis that they're perfectly healthy at 18.5, but then perish perinatally or postnatally. And the phenotype information that we gather from the systematic analysis at this stage, at the 18.5 stage before birth, is really valuable in many times in identifying why those mice don't make it. But in some cases, it still remains completely unknown. If you break this down into statistics, this is the overall percentages that we capture in this analysis. The largest percentage of mutants we find do die before the 9.5 stage, and there's a series of R01s that are now dedicated to understanding the role of those genes and do secondary phenotyping to understand the role of those genes for the pre-9.5 day lethals. The second largest class, of course, is those that perish after 18.5, and this is a really interesting class, we think, and the survivables fall into this category as well. Most of them survive until 18.5 and die sometime after birth. And the other classes you'll see represented there. So the challenge really with this type of approach is to identify ways to deliver phenotypic information to the community. So the individual defects in these embryos are very valuable in order to understand implications for human disease or even for developmental biologists to be able to begin to identify mechanistic relationships between genes and development. So to do this, our consortium group adopted a set of techniques, most of which what we're doing now has been pioneered by Mark Henkelman at the University of Toronto, but has been expanded through this consortium effort amongst the different groups. And one of the tools that we use quite a lot now is high-resolution micro-CT. It would be cost prohibitive and take a lot of time to section each individual embryo and provide traditional pathology type results for these phenotypes. But micro-CT allows us to be able to capture entire embryos with histology level types of data to be able to look for structural phenotypes and to have digital volumes that can be sent out to a variety of people and can be interrogated in a variety of different ways, as I'll show you. So this slide is just a pretty colored set of 3D reconstructions of micro-CTs of embryos at a variety of different stages. It goes postnatal to early embryonic, and you can see the features from the external views of what you can see for the embryos, but the value really is in the internal information that you can pull from this. So using a simple iodine-based contrast that's both relatively cheap and also relatively agnostic to different tissues, it gets soaked into everything but has variable contrast depending on the density. You can then look at the identify structural features with these embryos just as if you were looking at histology slides. And just to take you through this in real time where you can section through the different planes, an individual user can interact with this data very easily by being able to turn and move through in 3 dimensions the data to look for structural differences between mutants and wild types. And I think many of you who are anatomists in the room will easily pick out many of your favorite organ structures or different regions that you're interested in. And I think the legacy of this data will be realized many, many years from now when all of the different people with all the interests in all the different organ systems really can pour through the data and find value in it. Okay, so to curate this data and make it available to the public, which is a really important aspect of the program, there is, similar to what Steve mentioned for the adult phenotyping, a curation strategy through the Data Coordination Center to collect this data and also then to produce ways for individuals within the community to use it. So the culmination of the collection of the data and the QC, the comparison between our embryo data and adult data, all of that can happen through this system, and then ultimately users can pull up the data on a 3D viewer that might look something like this, where they can directly compare slices in a variety of different directions between mutants and wild-type embryos. And furthermore, the phenotypes that are identified, many of them are obvious and they're based on available ontologies, but some of the phenotypic information can be better gleaned through less biased approaches. And as a consortium, we're beginning to develop automated volumetric analysis and image registration strategies to be able to create an automated phenotyping pipeline. And these are especially good at identifying differences in volume that may be subtle to the individual who's reading the data, or other types of changes, but maybe not as good as a trained person at looking at large-scale dysmorphologies. Okay, so there's lots of interesting and obvious phenotypes that we have gleaned from these studies, and I'll just take you through some of the stories so that you get a feel for the kind of impact that this information has. So on the left is a gene called TMEM132, completely unknown. The only thing that's known about it is this transmembrane gene, but really interesting embryonic phenotypes. So you can see in these embryos at 15.5 days from the exterior that they're smaller, they have limb abnormalities, and they have spina bifida. So that's obvious from the outside, but when you look through the inside, you realize in the sections that they actually have duplications in kidney tissue. Sometimes they have kidney agenesis, and also have, not shown here, cleft palate and other types of internal abnormalities. Highly pleiotropic and a really interesting new find. The second one is GYG, and these are shown at the top of the slide on the right. GYG mutants have cardiac hypertrophy, cardiomyopathy, and I'm going to talk in more detail about these embryos in a minute. And SMDT1 is another gene with unknown function, but you can see it has excessive brain foliation in these embryos. These embryos mysteriously die shortly after this time point, and we can't quite figure out why. There's no structural defect in the heart that we can see, and we're not sure exactly what's responsible for the lethality. So a year ago or so, we published the first systematic analysis of the genes that we have been looking at in this context, and also a complete description of the pipeline and the information offered to individuals. And I just want to take you through a couple of high points from that study. First, as I mentioned, at the time that paper's based on the first 1,751 genes, but now that number has gone up considerably. But one thing clearly to point out is that the percentages between lethal, subviable, and viable are very consistent all through that time. We can look at how the types of phenotypes that are assessed at each of these stages in mutants, and we see that there's a large proportion at all stages in growth and cardiovascular development, but as you move through development to later stages, you see a greater involvement of phenotypes in different organ systems that can be very valuable to understanding disease associations. One of the interesting things that we found when analyzing the relation to human disease is that of our 399 lethal genes, 126 have human homologs with known disease associations. And in this case, at the time of our paper, 52 of these were the first report of a mouse model. So a really profound impact on being able to study these particular genes. And the same was true for subviables, 194 subviable genes and 57, oops, this got cut off a little bit, 57 human homologs had a disease association, and a smaller percentage of those were also the first known model of that. If you look statistically at our essential gene group and compare them to non-essential genes or all genes, there's a greater probability that you will find disease relationships within the essential group. So these are really important genes that play significant roles in development, but also are highly integrated into the disease landscape. And if you look across the different classes, viable, subviable, and lethal, and we characterize in some cases for the subviables both hets and homes, you find a real richness in the phenotype hits that you can recover from these animals, providing a lot of information that you can use to compare against disease profiles. In the percentage of disease association, the subviables are really a rich class where a lot of those genes, 66% or so, are associated with some type of disease. Okay. As we move on and consider the differences between mouse and human in terms of essentiality, studies have been done in cell lines to try to identify genes that are essential in human cells. So we looked at this group and compared them with the essential genes that we had recovered in the mouse. 35% of these could be compared, but 65% represented models where there were no mouse mutations yet. Of these, there was a very strong correlation between genes that were essential in mouse and genes that were essential in human. We recovered 19 human essential genes that were common to all three studies that were non-essential in the mouse, but only in 3 cases could we confirm that this was really a null phenotype. So I think there's still information out there to try to determine the overlap in these. But nonetheless, a very strong correlation between mouse genes that are essential and human genes that are essential. And this further bears out, I think, from Terry Meehan's and Damian Medley's paper in Nature Genetics, and poor Damian's going to have nothing to talk about tomorrow, I don't know. But we're very grateful for this work because it illustrates something I think really important about assessing these essential genes. So our genes result oftentimes in mice that are not born. The essential genes actually kill off the embryos before they're able to see light. So the question really arises about how is this going to inform in human disease. But in many cases, and what this study showed, is that there are a lot of genes that where the autosomal recessives do have lethal type phenotypes that are similar to what we see in the mouse, but there has to be a little bit of extra work in comparing these. So, for instance, mice die at birth for a variety of reasons where humans would not suffer lethality in those cases. So, for instance, cleft palate, there's no NICU in the mouse room that rushes these little pups off and treats them and is able to help them recover from this disease. So when you start comparing apples to apples in that sense, you see a strong correlation between the autosomal recessive genes that we see lethality in mice and things that are really detrimental to early postnatal viability in humans. And so that's, I think, a really interesting finding from this study related to essential genes. The other thing that's really that you can consider is in looking at many of the genes that have disease associations, the opposite is true. So we find that while the gene is lethal in mice, there are diseases where even adult humans suffer from loss of function diseases where there are adult human phenotypes. And in some cases, this is because the human loss of function alleles are less severe due to redundancy or compensation by modifiers. So the robustness of the human genome supports a further development. And some cases where lethality from recessive null alleles in mice are correlated with hypomorphic or neomorphic alleles in humans, but the information that is provided from the phenotyping in these cases is still very informative. And I'll just take you through a couple of examples. So one of the things that my group is interested in is cardiovascular development and genes required for cardiovascular function. And we've spent a lot of time developing live imaging techniques, and that's shown on the right-hand part of the slide, looking at embryonic heart function. This is not something that you would do as a primary tool in screening, but is very valuable for secondary analysis. So we wanted to advance beyond our phenotyping and come up with strategies to be able to to pull out information about early cardiovascular development in an advanced way as a primary tool on the screen. So we enhanced the micro-CT procedures to go back and look at embryos within the implantation site. And so the one on the left was still within the deciduum, but from this information you can capture early development of the placenta and hallmarks of vascular development that actually inform the level of circulation and the types of heart function that you have within the embryo. And by looking at these, the extraembryonic vasculature in this way, we can capture that information as part of the phenotype and begin to then understand how those phenotypes would relate to human diseases. And one example that I want to show you is ALG10B. So null mutations in ALG10B result in lethality between 9.5 and 12.5 days in mouse development. And you can see the phenotype shown here. And one of the interesting aspects of this phenotype is if you look at these extraembryonic tissues, the yolk sac that surrounds the embryo, you can see abnormal vascular development. So the wild type is on the top and the mutant is on the bottom all the way to the left. You see a lack of that vasculature that surrounds the embryos. And there's other defects within these embryos as well. The interesting thing about this is that the human disease that this is related to in humans actually is a reduced susceptibility to a drug-induced long QT syndrome. So it still affects cardiovascular function, and that's a point mutation. So it'd be very interesting to model that point mutation with respect to this null allele, but the homozygous null affects early heart development. So again, same system, slightly different manifestation depending on the mutation. It's really an opportunity, as David mentioned, of looking at very precise disease-related alleles and comparing them directly to null information. The second gene that I want to talk about is glycogen storage disease, is a gene responsible for glycogen storage disease 15. It's GYG. The mouse has a single gene, humans have two. And the patients that suffer from glycogen storage disease 15 have a skeletal muscle myopathy, they have muscle weakness, and the glycogen storage disease 15 patients also have arrhythmias. Interestingly, this gene is also mutated in a second disorder called polyglucosan body myopathy 2. This has skeletal muscle phenotypes but no cardiac implications. We looked at the mutant for this gene in glycogenin. LAXI expression shows very strong expression very early in the embryo and is localized to the heart. Beautiful heart-restricted expression that continues, and then we see expression surrounding the smooth muscle cells around major arterial segments, and the middle panel shows the expression around the umbilical artery. Homozygotes are found in normal proportions at 18.5, but they die shortly after birth. And they exhibit a cardiac phenotype, cardiomyopathy, as I alluded to earlier, where you have this enhanced thickening of the ventricular walls. But there's other types of disorders associated with these mutants that are not generally thought about with respect to glycogen storage diseases. For instance, the thymus is just enormous. So you can see it in the wild type image. The thymus is brightly stained nodules just above the heart. In the left-hand side image, where the number 3 is pointing, are the thymus in our GYG mice, and they're about 4 or 5 times the size of the wild type thymus. We don't know yet what accounts for this difference, but this is a phenotype that I think many have not considered. The heterozygous mice in this case do confirm the the arrhythmia type phenotype seen in adults. These mice have an increased susceptibility to atrial fibrillation when you conduct pacing experiments. So we think that the heterozygous phenotype actually matches pretty well with the arrhythmia data from human patients. And then finally, we see another phenotype within the homozygous embryos that's actually completely unexpected. And that is that the spinal cord of these embryos has a series of breaks in it that we think results from neural degeneration. And you can see in the images on the left, as well as in the middle panel, and to a small degree in the transverse sections, these gaps or spaces within the spinal cord. And we believe that that's where cells have degenerated during development. We don't know how that contributes to lethality, but one of the interesting things is that there have been variants picked up in the second GYG gene in humans, GYG2, where patients have severe neurological disorders. Now, this is a variant of unknown significance, but now with the mouse data, it's something that provides an opportunity to look at further. Okay. Finally, I just want to say that we're very fortunate to be at Baylor College of Medicine, where there's such a large number of robust activities going on that relate to human genetics and genomics. And it's an opportunity for us to be able to have really direct conversations with many groups that are interested in identifying variants related to human disease. And it provides an opportunity for those genes to enter into our pipeline, to have an expedited strategy, to be able to create null alleles that can inform these studies. So the real-time flow of information in that respect is really valuable. And in addition, we've been embarking on pilot studies to begin to model precise disease alleles and utilize the infrastructure from the phenotyping pipelines to be able to provide phenotype information to the community. And that's a very valuable exercise, and something that we would like to see become more of a part of the overall landscape of many groups. Finally, I'll just acknowledge the people who have been involved in these studies. There's a long list of people here at our site, as well as I echo the acknowledgments that Steve made to all of the members of the IMPC consortium. This is truly a group effort with a lot of value imparted by a large number of people across a lot of countries. So thank you very much for your attention.